Hello, my name is Hossain, and I will be going over cockroach aggregation behavior and gut bacteria. My presentation is made up of why I chose the topic, an introduction of the key terms, a summary, a take-home message, an honorable mention, and of course the specific paper in mind. The paper being Succession of the Gut Microbiota in the Cockroach Bactella germanica. So what inspired me? We are currently discussing cockroaches in our microbiology class, and I remembered how interesting of an animal they were. Amo, Hikma, and I had created an experiment last semester that evaluates whether cockroaches can detect and prefer mates of their own species. Our hypothesis had been that the P. americana cockroach would have a preference between P. americana and a B. germanica. More specifically, they're conspecific, meaning of the same species, rather than their heterospecific, meaning of a different species. We tested this theory over a 4-day period in which we observed 12 animals. One bin was used to hold the Periplaneta cockroaches, while the other was used to hold the Bactella cockroaches. A larger bin was used to observe the animals' preferences, almost like a makeshift arena. This larger bin was divided into 3 sections. On the right, a P. americana was placed under a red cup with holes, and on the left, a B. germanica was placed under a smaller cup with holes. This was done to transmit the pheromones of the animal. In the middle, another P. americana was released to represent the cockroach's first choice, which was either left or right. A tally mark was recorded as illustrated by the graph. Unfortunately, our hypothesis was proven incorrect due to a lack of insignificant data. But nevertheless, this experiment inspired me to take a deeper look into the behavior of cockroaches as a whole. However, instead of the P. americana, today we will be focusing more on the Bactella germanica. The Bactella germanica is a German cockroach, also known as the cotton bug. It is a species of small cockroach, typically about 1 centimeter long. It is distinct in the way it has 2 dark, roughly parallel streaks on the pronotum running from behind the head to the base of the wings. These insects are known for carrying pathogens that can contaminate food and produce allergenic proteins that cause human illness. In fact, common diseases German cockroaches carry and spread include salmonellosis, dysentery, and gastroenteritis. If spreading disease wasn't enough, German cockroaches also spread allergens into the air with their fecal matter and shed skin. You'll notice that fecal matter will be discussed again at some point in the presentation. Also, I must mention that before moving on to the experiment itself, a few key terms have to be properly defined and understood. Fecal aggregation agents, for example, is another word for pheromones. Pheromones are chemicals that affect the behavior of other individuals of insects. Studies show that the gut microbial community contributes to the production of these chemicals. As for aggregation, it is when a group of organisms of the same or different species live closely together, usually in clusters. Moreover, aggregation pheromones are emitted from the animal in order to attract individuals of the same species to locations for specific reasons, such as feeding, mating, and so on. Finally, the microbiome is the collection of all microbes such as bacteria, fungi, viruses, and their genes that naturally live on in the bodies of all animals. In fact, these microorganisms inhabit an animal's gastrointestinal tract to get a better understanding of the microbiome as a whole. I've included a short clip that sums it up in layman's terms.
Our bodies are home to over 100 trillion microorganisms. These microorganisms are found everywhere and make up microbiomes. Over the course of evolution, microbes have evolved with us and have an important role in the human body. Without our microbiome, we wouldn't be able to survive. Microorganisms live all over the body, but have the highest density and diversity within the gastrointestinal tract. The gut microbiome is vital for digestion and helps us break down complex carbohydrates, such as starch from potatoes and cereals, and fiber from fruits and vegetables. The microbiome also helps provide vitamins and minerals needed to survive. As well as aiding in digestion, the gut microbiome is thought to play an important role in protecting against disease. Stomachs with a healthy microbiome are thought to be so diverse that when pathogenic bacteria are present, such as Helicobacter pylori, which causes stomach ulcers, they aren't able to thrive and cause disease.
Now that you have more of an understanding on the material, let's take a closer look at the paper by the Institute of Biodiversity and Evolutionary Biology. The article begins by defining the bladobacterium. Cockroaches harbor this endosymbiont in bacteriocytes, which are specialized cells in the fat body that are acquired for host fitness and fertility. Genome sequencing revealed that bladobacterium plays a role in the host nitrogen metabolism and in the synthesis of essential amino acids. In Blatella germanica specifically, bladobacterium enters the oocyte plasma membrane during ovarian development. Once inside, it participates in yolk utilization, thus contributing to an embryonic development. All that to say is, in this study, researchers examined this bacterial load and the changes in microbial diversity both in the embryos and in the gut of the B. germanica in each of the 5 nymphal instars, as well as the adult males. The findings shed light on the mode of gut microbiota acquisition throughout cockroach development. On the right, we see a figure that illustrates the time course changes in gut bacterial load throughout the development of Blatella germanica as assessed by qPCR test. Bacterial abundance was inferred as the copy number of bacterial 16S RNA genes in total DNA extracted from cockroach guts. As shown, 4 determinations were carried out at each stage. In addition, bacterial load increased from stage N1 to stage N2 and then remained relatively constant throughout the following moltings until the adult stage. Moving on to the experimental design, the experimental B. germanica colony originated from a stable laboratory in Barcelona, Spain. The insects were then bred in lunch boxes, which were renewed weekly. 40 cockroaches were maintained per box. The nymphal instar, otherwise known as the young developmental stage, was identified by measuring head width, whereas adults were identified by their wings. The animals were then labeled accordingly. During dissection, 2 specimens from the same brood were selected as biological replicates for each of the developmental stages. The ootheca, otherwise known as the embryo contents, were extracted by drilling the external surface of the insect with a pipette tip and sucking out the embryo-containing fluid. Surface samples were then routinely tested for bacteria by PCR. Absolute quantification of bacterial 16S rRNA gene copies were carried out by quantitative PCR using the universal bacterial 16S rRNA gene. More specifically, 4 measurements per stage were used to statistically test the change in bacterial load from one stage to the next. This step is vital because the composition and the structure of the gut microbiota in each sample were characterized using 16S rRNA gene amplicons. Using this data, a cluster analysis was then used to study intersample similarity in taxon composition. Furthermore, the guts of 3rd instar nymphs and adults were inspected by scanning electron microscopy. I should also mention that before dissection, the cockroaches were starved for 3 days to obtain a clear gut surface. In the end, all sequences obtained in the study were submitted to the European Bioinformatics Institute. Results. The statistical approaches implemented show that part of the variation in gut bacterial composition in B. germanica is stage-related, and that N3 nymphs and adults differ from the other stages. Moreover, bladobacterium was the only bacteria detected in embryos inside the ootheca. Overall, the gut content among most nymphs is similar but different from the adults as shown by the pictures. This similarity suggests that at these stages, the gut habitat is suitable for a specific fraction of early colonizing microorganisms. The main increase, both in bacterial quantity and diversity, takes place after the first molt, after which bacterial load and richness remain constant. According to these results, it is more likely that the gut microbiota is horizontally transferred via fecal contents rather than vertically via transfer the egg. Overall, little is known about the mode of acquisition of gut bacteria or their ecological succession throughout the insect life cycle. Nevertheless, the study reports on the bacterial taxa isolating from different developmental instars of the cockroach Blatella germanica. As explained earlier, the bacterial load in the gut increased 2 orders of magnitude from the first to the second nymphal stage, coinciding with the incorporation of the majority of bacterial taxa, but remained similar thereafter. In summary, bacterial community composition in the B. germanica gut differs between nymphal instars and adults. Specifically, the former has an additional abundant genus, Bacteroides, that decreases in abundance and when the insect molts as it becomes an adult. An honorable mention. As stated in the beginning of this presentation, we are currently discussing gut bacteria and cockroaches in our microbiology lecture, which encouraged me to adopt a similar approach to my topic. Researchers investigated whether gut bacteria contributes to the formation of fecal aggregating agents in the publication Gut Bacteria Mediate Aggregation in the German Cockroach. The findings showed that the symbiotic gut microbiota contributes to the creation of volatile carbolic acids, a form of pheromone, that operate as fecal aggregation agents, and that cockroaches can distinguish between the complex scents emitted by a diverse microbial population. This is significant because this article refers to both my animal behavior project and the paper described in my presentation. Animal behavior in the form of pheromones and how they influence behavior, as well as my lengthy discussion of aggregation and its significance. It's fascinating to see how in-depth one can get while studying cockroach behavior. As a result, several studies on these animals have yielded similar results. Thank you.